Hello, this is group 2, and we're going to be talking about the codon and anticodon. The codon is a triplet of nucleotides on the mRNA strand that code for a certain amino acid. If we take a look at the genetic code, we can see that 3 different nucleotides can make up an amino acid. For example, if you look at the last box, if we have a G, G, U, that triplet is going to code for a glycine. And as you can see, you can have a difference on the third nucleotide. So instead of a U, you'll have a C, and that will still code for the same amino acid glycine. But in general, we use this table to code for an amino acid if you have a codon. So the anticodon is a complementary to that codon, and it is located on the tRNA. The tRNA, which we have a picture here, recognizes specific codons on the mRNA strand and carries its respective amino acid. So, like I said, this is a tRNA. Here we see that it's carrying the amino acid at the top. And at the bottom is the anticodon loop, which has the anticodon composed of 3 nucleotides. So how does this all work? So how does it actually work? Well, starting from DNA, you have the coding strand, which is what we want to translate in the end eventually. We find the complementary strand that is antiparallel to the coding strand, and eventually that will be transcribed into the mRNA. Again, it's going to be antiparallel to it. So here we first have a codon of 3. And the tRNA, what it's going to do is going to bind the codon and the anticodon complementary to each other and antiparallel. So if it's going from 5 to 3, this will be 3 to 5. So if it matches, it pairs the first codon anticodon, and then it will release the amino acid to a growing chain of polypeptides on later codons.